to another sessions of the Faculty of Pharmacy Universitas Indonesia Virtual Summer Course 2022 Advanced in Pharmaceutical Science. Before we start, please let me introduce myself. My name is Donna Mareta Aryastanti, a faculty member of Faculty of Pharmacy Universitas Indonesia, and I will be your Master of Ceremony for this virtual summer course sessions. And first of all, I would like to greet our honorable guest for this afternoon, Dr. Apoteker Fadlina Chanisa Putri, Magister Science, the Vice Dean for Academic, Research and Student Affairs, Faculty of Pharmacy, Universitas Indonesia, Dr. Swarit Jasial from Data-Driven Chemistry, NICE, Japan, the honorable speaker for today's lecture, Apoteker Rezi Riyadi Shahdi, Magister Science, the honorable moderator for from the Faculty of Pharmacy, Universitas Indonesia, the honorable professors, lecturers, and all faculty members who attend today's lecture. Last but not least, I also would like to greet all audiences and participants. This summer course is aimed for students and fresh graduates who are interested in increasing their knowledge of pharmaceutical science with a broader perspective, because we invite professors and lecturers from Universitas Indonesia and well-known universities abroad. And on this afternoon, we will have the sixth lecture from Dr. Swarit Jasial with the title Chemoinformatics in Silico Techniques Applied to a Range of Problems in Chemistry. But before we begin the lecture, please allow me to read the profile of this session's moderator. Apoteker Rezi Riyadi Shahdi, Magister Science, has been a lecturer since 2012 at the Biomedical Computations Lab, Pharmaceutical Chemistry Divisions, Faculty of Pharmacy, Universitas Indonesia. He obtained his Bachelor of Pharmacy at Universitas Indonesia in 2010, and he obtained his Master of Pharmacy at the same university in 2012. In 2022, he continued his studies to a doctoral degree and is currently studying as a PhD student at the Data-Driven Chemistry Lab, Nara Institute of Science and Technology, Japan. His research is in the areas of medicinal chemistry, in silico screening, and phytochemistry. All right, without further ado, please welcome our honorable moderator, Apoteker Rezi Riyadi Shahdi, Magister Science. To Apoteker Rezi, the screen is yours.
Hello, everyone. Thank you, Master of Ceremony, for your brief introductions. Good afternoon, everyone. My name is Rezi, and I will be serving as your moderator for today's lectures. And the title of this lecture is Chemoinformatics and Silico Technique Applied to a Range of Problems in Chemistry. Before we begin the lecture, please allow me to read Dr. Swarit Jasial, curriculum vitae. Please, the committee, can you show the slide? Okay, thank you. And this is Dr. Swarit. Bachelor. He has Bachelor in Bioinformatics in JP. Sorry if I pronounce it wrong. JP University of Information Technology, India, since 2007 until 2011. And then he got his master degree in Life Science Informatics degree. Please, next slide. Next slide, please. Yeah, okay. And master degree in Life Science Informatics degree from University of Bonn, Germany, from 2012 to 2015. And maybe he can speak German a little bit, maybe. And then next, oh yeah. And then he got his PhD in computational life science or chemoinformatics. Maybe this is hard for me to read. Bonn Aachen International Center. International Center for Information Technology, be it Bonn, Germany, from 2015 until 2019. And then currently he is working as my assistant professor in the data-driven chemistry lab at Nara Institute of Science Technology, or NICE, Japan, since July 2019. Currently, this is my campus, our campus. Currently, his research focus is working on target identification for natural products and also predicting antiviral activity of odorants in collaborations with some corporation of Japan, Kao. Maybe it is also popular in Indonesia, Kao, especially for women. He is also actively supervising a PhD student in his lab with research project focusing on prediction of activity clips, where the interpretability of machine learning models is explored to make them chemically intuitive.
Tapi kalau ini udah kayak
He is also investigating computational screening strategies for compound using two-dimensional and three-dimensional molecular representations. All right, without further ado, please welcome our honorable lecturer, Dr. Swarit. The time and screens are yours.
Thank you very much for your kind introduction. So let me share my screen first. I hope it is visible to all. Okay, so good afternoon, everyone, especially in Japan and Indonesia, and whoever is joining from different parts of the world. And thank you for joining the virtual summer course 2022, that's Advanced in Pharmaceutical Sciences at Faculty of Pharmacy, University of Indonesia. And so let me start with the topic for today, that is chemoinformatics. And as the title suggests, it is a blend of chemistry and informatics. So it means the application of informatics or computers in the field of chemistry. So the outline for my presentation today would be a brief introduction, or it would be more related to different aspects of chemoinformatics. So I'll start with the drug discovery process, and then I would focus on different aspects of chemoinformatics, followed by molecular representations, how we represent the compounds in the computational form. And then I would focus on data mining and machine learning methods, which would include some mathematics or mathematical models to predict activity of compounds. And finally, I would like to present about 2 case studies for the practical application of chemoinformatics. One would focus on the regression analysis that on the topic prediction of antifungal activity against fungi called Aspergillus fumigatus. And one would focus on machine learning for distinguishing pan-assay interference compounds, or so-called PAINs. So let me start with drug discovery process. So generally, the drug discovery process goes like this. You are focused on a particular disease which has a particular drug target, and which the drug candidate generally focuses on. And basically, in this part, we want to find a drug molecule that is specific against a specific drug target. And once that drug candidate is found, there are certain protocols such as animal studies, clinical studies for studying side effects, and so on. And it takes a long time for a drug to be marketed and come to the markets, such as this drug called aspirin. And so drug candidate is also known as ligand, and this ligand compounds binds to a biological target, which can be either protein, enzyme, or a certain receptor. And in this way, it either initiates the process or inhibits. If it initiates, it's called as agonist, and if it inhibits, it is called as antagonist. So the main focus of the drug discovery process is drug specificity, and traditionally it has been focused on finding a specific drug against a specific target, as I told you in the previous slides or one drug molecule against one target so that it has high potency and selectivity. Now, what is this ligand and protein? So basically we want to find, for example, you have this ligand, it is in the yellow color and binding site. So there are different different types of interaction which a ligand undergoes with the binding site. For example, non-covalent bonding here. And this is an example of a drug molecule, and this is how the target is composed of. For example, you can see it has a binding site where this drug tends to bind and then produces a certain effect or certain action, which can be either agonist or antagonist. So it's kind of a lock and key arrangement, because there is different structure or conformation of a ligand which is complementary to the space defined by protein's active site. So you can imagine it ligand as a key and binding site as a lock. So there should be favorable interactions between ligand and the side chains of amino acid in the active site, like electrostatic interaction, hydrogen bond or hydrophobic contacts, or so on. And there are different classes of drug targets which are mentioned in this pie chart, like enzyme, nuclear receptor, DNA, ion channels, hormones, and there are different types of these drug targets which are used in current therapies or so on. Now, one of the aim is to find structure activity relationship. So there are different substructures which are present in a compound, as you can see, for example, OH, or you have a carbon in the ring or so on, which are labeled in the right side. And certain substructures, the combination of substructure might lead to enhancing the activity of this compound against a particular target. However, there can be certain substructures which do not contribute for favorable interactions, which might lead to inactivity. So this relationship between structure and the activity of compound is generally a focal point in chemoinformatics. Now we want to find basically the best fit molecule, and you can see this is suppose the active site, and there are different small molecules in the which we want to find. So we want to find like a tight fitting complex. For example, out of all the colors, green one fits the green one, suppose. So we want to find that best fit molecule which leads to favorable interaction, and we have a large number of molecules in the library. So it's an example, like in English we say finding needle in a haystack. So you have a haystack which consists of large number of molecules, and you want to find the best molecule out of this lot. And this is tested in different type of binding assays, experimental setups, or so on, so as to find the best fit molecule. And one of the techniques which is used to for screening of compounds is high throughput screening, which is a method of scientific experimentation that comprises the screening of large number of compound libraries against many biological targets. So you can imagine millions of compounds on the rows, and hundreds of assays or different experimental setup against different targets. And this way, in a pharmaceutical or a wet lab setup, they test each and every molecule against so many different targets or so on. And they measure different there are different ways of measuring the activities, and these are some of the common activity measurements which are used. For example, IC50, which is how much of a particular inhibitory substance, or in our case ligand or drug, is needed to inhibit a given biological process by 50%. And there is another one called as the dissociation constant, or called Ki, which describes the binding affinity between the inhibitor and the enzyme. And there is another measurement called as minimum inhibitory concentration, or MIC, which is the lowest concentration of a chemical which prevents visible growth of a microorganism. It is generally used for bacteria, fungi, etc. And the lower the value of IC50Ki or MIC, which means a lower amount is needed for particular drug or a compound to inhibit the activity and therefore inhibit the process, and therefore it's the higher activity in this case. So these values should be lower for higher activity. Now in wet lab drug discovery process, as I told you about high throughput screening, so millions of compounds are screened against so many different targets. For example, 10 raised to power 6 compounds are screened and you get around, for example, 1,000 actives in this experimental setup. And however, these kind of experiments which are done on a large scale are generally have high risk of false positives because they are mostly automated and sometimes they are not so efficient because there can be contamination or so on. So a large scale experiment generally is not advisable, but this is how the way things work in pharmaceutical or a wet lab setup. And moreover, just by screen, there are many factors which influence the activity, such as purity of the sample, the mechanism of action, which activity values, scaling of activity values, or so on. And therefore, after doing all these processes, out of when you screen such amount of compounds in, and you have large number of resources used, you only end up with 1 to 10 hits, and then hit to lead optimization when you do by looking at the analog synthesis of the compounds and looking at this different properties such as ADMET properties, absorption, metabolism, excretion, and so on, and different pharmacokinetic properties. You end up with 0 to 3 lead series of analogs, and then out of them, when you test in clinical trials or so on, you end up with 0 or 1 candidate. Sometimes there is a failure of drug discovery experiments after utilizing so many resources and cost. And as you can see, there are many factors which are which have the failures of drug development, and this is one of the pie charts showing them, such as even if you find them active or so on, they end up failing in, for example, animal toxicity or absorption in the body, or like different side effects in the human beings and so on. So after doing such large and huge amount of experiment, you end up basically with loss of cost and resources in this case. So it is very important because this drug discovery process is a long process, and therefore there are different phases of this discovery or drug development in this case. And therefore it is advisable, or I would say it is efficient to find these predictions of failure in early stage of drug discovery rather than when, because as you move to further towards the later stage of drug discovery, the cost increases from, for example, you can see this graph from 10 to 12 years up to 1,000 million or so on. So it increases in the later phases of drug discovery. So why not predict the failure of drug discovery process in early stage? Because, and now and the present case, because the number of compounds are increasing year by year, and the increase of data in different databases or so on has been exponential over the last few decades or so on. So and it is predicted that this would go exponential. So we have a large amount of data which we can utilize for the prediction of failures in early stage of drug discovery. We can utilize the knowledge contained in the data in that case. And this is where chemoinformatics come into place, and therefore we utilize the data, we make the predictions from the data, and we predict the failures of the compounds in early stage so that there are less number of compounds or very few reduced number of compounds which are tested in the laboratory, so that we can we don't use all resources, time and cost. And this is why we use computational methods for prediction of activities of compounds or potency of compounds against a specific target or so on. So now we move from in vitro to in silico plus in vitro. So in vitro, diverse set of molecules are tested in lab and time and resources are huge. But in silico plus in vitro reduces the number of compounds which are tested in the lab based on the prediction of drug likeness, solubility, binding, pharmacokinetics, toxicity, and different side effects using computational techniques, so that time and resources are reduced. And so now the definition of chemoinformatics, it is a field of information technology that uses computer programs to facilitate collection, storage, analysis, and manipulation of large quantities of chemical data. So we use computers for the prediction of certain properties of compounds. And these are different aspects of chemoinformatics, which I would focus on in today's lecture and also in my case studies, which I will present. And that would include the databases, because a huge amount, as I told you, huge amount of data is being generated and is being supplied to different publicly available databases as well as commercial databases. And therefore, the maintenance and storage of this data is a very important aspect of chemoinformatics. Second aspect is machine learning. Like, we use computational models like decision trees, neural networks, self-organizing maps, etc., on the molecular data, so as to predict different properties of compounds before they are tested in the lab. And how drug-like are these compounds? So we use certain kind of experiments, in silico experiment like virtual screening or chemical biology networks, etc., which is also one of the important aspects of chemoinformatics. So now we come to how these chemical compounds are represented in the computer form. So because I think all are here from the pharmacy background, so they are more familiar with different type of organic compounds. So for example, phenylalanine is the basic nomenclature. Then you can represent a compound in 2D structure. You can also include conformations or using 3D coordinate as the 3D structure. And also there are different surfaces involved with the compound, like van der Waals interaction or so on. So this is a hierarchical scheme of representation of a compound in different way. So there are a lot of information contained in a compound structure. One of these basic informations are, for example, atoms present. What are the atoms present in a compound? For example, in this you can see there are 8 carbon atoms, 9 hydrogen atoms, nitrogen, and 3 oxygen. Then next, what is the connection between the bonds? What are the different bond types? There are single bond, double bond, there are aromatic rings present in the compound. Also, there is stereochemical information, which and there are different sometimes charges available or the compounds which are shown in the as in here in case of nitrogen. There can be several isotopes present in the compound. And finally, what is the conformation of the compound and what are different conformers of the compound? Because at the end, the compound is a 3D structure. So this all information we have to represent in the computational form. And therefore, we move from chemists to the representation. So this 2D, 3D model chemical structure, we convert it to an intermediate representation, and finally to an internal machine representation, which is understood by computers. And there are different type of ways in which a compound can be represented structurally, and this can include a simple string-like representation called as line notations, or it can include large amount of information in a file like a connection table. And so these are various types which I would be telling you in my further slides about these different structural representation of molecules. So first of all, let's consider the graph theoretical representation. So it basically tells about the information about molecular topology. So graph is like, you can imagine graph as a set of nodes connected by edges, simple to like a graph. As you can see here, the chemical structure of aspirin, it can be represented in the form of a graph where the nodes represent the atoms and edge represent the connection between them. And with this information, we can form a connection table, which consists of atom and bond blocks. And this atom information, or the nodes, they are different relative 2D atom positions that can be inferred from the connection table, assuming standard bond lengths and ignoring conformal stereochemical information. So, for example, a 2D compound can be represented by two-dimensional Cartesian coordinates, and for 3D you can have three-dimensional Cartesian coordinates. And in this way you can preserve the information of the compound, like atom table store additional information about element type, stereochemistry, charge. So there are different blocks which can be present in the atom tables. And in chemoinformatics, usually the representations which we find, mostly they have hydrogen atoms, as you know, most of the chemical compound. And these hydrogen atoms are implicit because the representations we will discuss consider them as implicit, or they automatically calculate. And we usually represent the chemical structure drawing as hydrogen suppressed ones, as represented in this case in this example shown. So a simple compound can also be represented in the form of bond table, which shows a connection. So so you can see there are different labels or different numbers given to different bonds. So it's a unique identity for a different bond, and bond table can represent such as this, where node A is connected to node B. So 1 and 2 are in the same row, and similarly 1 and 6 are in the same row. So in this way, all the connections are generally represented. So this 2 and 3 are represented by bond table. And a further annotated bond table could be that what is the bond order of the connection. For example, here in benzene, you can see this benzene structure can be represented as an annotated bond table, where you can add an additional column of bond order. And so in this way, each edge can also be connected with a property. Here, for example, for 1 and 2, the bond order is 2. So we know that it's a double bond. And this way, this bond table represents information of this chemical structure. And a connection table for molecules would further be a combination of the Cartesian coordinates and as well as the bond block. So you can say an atom block and a bond block, and you can see the corresponding between 1 and 2, the carbon atoms represented here and their coordinates. And this can help us in the reconstruction of the compound in computational form. So these connection tables are generally very large memory consumption tables, because they have they contain huge amount of information, not only atom, bond, but also property information like charges and so on. And these files are like huge. For example, this is an for alanine, this information is shown here, but so many bond block, bond information for each and every atom, as well as bond contained in this compound. So imagine for a database, how much information will be contained in such a huge file of connection table. This was developed by one of the company MDL. That's why it's named as .mdl format, but it contains a large amount of information. And for a database of million compounds, it would be hard to store and it requires a large amount of memory. So therefore, we need even simpler form, because these, although they contain all the information in these MDL files or connection tables, but they are very complex as well as memory consuming. So we need more compact representations for that case. And then we come to the linear notations of the chemical compounds. And linear notations use alphanumeric characters to encode a molecular structure. And there are different types of linear notations. One is called as SMILES, and that is simplified molecular input line entry specification. And INCHIS, which is IUPAC International Chemical Identifier. So SMILES is a small string-like representation, which I'll be discussing further in the slides. And INCHI is also a string, but it is like a unique identifier, because for tautomers, etc., it is generally a unique index, and you can, for each and every tautomer or so on. However, SMILES are different, but generally SMILES are easier to read and easier to represent. So we, in the majority of the publicly available database, the both SMILES and INCHI are given, but SMILES are generally preferred to further pre-process the data or so on. So let's look into the SMILES format. And SMILES format, this is a very simple example of SMILES format, which is like carbon monoxide, and that is, as you know, it's hydrogen suppressed. So it's a simple structure CO, and you don't represent hydrogens in this case. Next, you use branches, because you can see you select the longest chain. It's similar to, and then you it's similar to like we represent in organic chemistry, but yeah, it can be nested or stacked. So we can represent the longest chain and then branches in the brackets. For example, this compound, and you can see after the longest chain, this methyl is like the branch, or so on. Also, you can represent rings, like generally, if you have a ring and if you have aromaticity, like double bond, conjugated double bonds or so you can represent using lowercase characters or so on. And it's simply represented with a number, like where the ring is closed, like C1 here in this case, and here the ring is closed. So you represent it with a number followed by the longest chain. So these type of representations are usually contained in the different databases. So I would now talk about the storage of data in different databases, as it is also an important aspect of chemoinformatics, because we have huge amount of data available. And one of the databases which we generally use is ChemBL, which is maintained by European Molecular Biology Institute, EMBL, and it has bioactive compounds, small drug-like molecule. So it only contains their activity in different measurements like IC50, Ki, or so on, which I talked about earlier. And these are deposited by with the help of literature, which compounds published in literature, medicinal chemistry literature, or so on. And there are around 2.2 million compounds in this databases, which are and have information of these compounds against 14,000 targets. So 15 million ligand target interactions. So this data is quite useful when you want to make activity predictions or so on. And it's generally very beneficial in whatever projects we use in chemoinformatics. Another database which is common is PubChem, which contains a large amount of compound, 3.4 million tested compound, but it has the assay information of the compound. So you basically have the compound and its tests against different types of assays and different experimental environments. So it's labeled either active or inactive based on different potency measurements. And so if you want to find, for example, inactive compounds, then this is a very good source because it provides you information about inactive data for a particular target or so on, as the assay profile is maintained in this database. So it has around 3.4 million tested compounds against 11,000 targets. And next, we have around it has around 237 million activity annotation, which is a huge network. And this data can be very useful for making predictions or so on, which I'll talk further in my presentation. Another database which is popular is Drug Bank because it contains information about the approved drugs or the drug candidates which are already approved by FDA or so on, the Food and Drug Administration. So you can see around 2,528 FDA approved drugs or small molecule drugs are present here, and around 5,000 protein sequences are given for these against these drugs. So it's a very useful information if you solely want to focus your research on a drug related entity. So these are different types of databases. However, there are many other databases, but these are the most common ones which we generally use in our research in chemoinformatics. But there are also like Zinc databases, very popular, and which has a lot of compounds which are generally used for random search or so on. So now coming on to the computer-aided drug design, and as I already told you about the storage and the amount of data which is available. So now what to use, how to use that data, and how can we conduct this computer-aided drug design? And there are different aspects of that. One is structural based drug designing. In this structural base, it is one should know about the target or the high resolution protein structure of the compound. So the target is known here in structural based drug design. One of a popular example of it is molecular docking, where you try to find score of the compound against a binding pocket or so on, and then you see its binding affinity, whether it's interacting with amino acid residue or so on. You can see a particular target structure. However, it has a general limitation because many drug targets do not have experimentally determined structure. Although protein data bank is a very good source of crystal structure and different binding conformation of ligand and targets, but however not all targets are available because they are determined experimentally through NMR spectroscopy or so on, and it takes a lot of time to find this crystal structure. So not much data is available for the targets, but still many different studies are going on with whatever the targets are available. And the other aspect of computer-aided drug designing is ligand-based drug designing, where we don't know the target structure, but we know about the active compounds, we know the structure of the compound. So we focus on our research based on the compounds we have in our hand. And we focus mainly on the molecular similarity method. And we focus that the similar molecules are likely to have similar properties because they might contain similar combination of features which are responsible for activity against a particular target. And there are many research which focus on ligand based too, and these are like prediction of molecular properties from similar analogs or so on. And so this type of study doesn't require the knowledge of protein targets, but you can find information from the substructural features present within the compounds. But you should have a good amount of data for a particular disease or a particular target you are doing, and which you can fetch from the publicly available databases. So my domain of research is generally ligand-based drug designing, and I mostly focus on this part in my further slides. And so as I showed you this slide previously, that we move from chemists to computational representation. So I showed you intermediate representation, which is the text form or the string form of the compound. This is miles or inches in that case. However, the computers, they understand binary language, which is like machine language of zeros and ones. So we need to convert our compounds from this intermediate representation into binary representation, which can be understood by different mathematical models or computational models. And this is where the concept of molecular fingerprints come. Fingerprint, as you know, fingerprints are unique for a person to person. So we can also make molecular fingerprints, which are like combination of 0 or 1 or bit string, which are unique identifier to a particular compound. So every different compound will have a different set of bit strings or feature lists. That's why it's called as molecular fingerprint. And one of the example of molecular fingerprint is Max fingerprint keys, which is very basic example of molecular fingerprint. So suppose we have this compound here, and we have a bit string vector of 166 bits, which are defined already. For example, double bond O is present in the first feature, and this substructure is present on the third. This ring is present here, and this ring with sulfur is present here. So we want to represent this compound. So we would look into the substructure, and if it is present, we would label this bin as 1, and if it is absent, we would label it as 0. And for example, this double bond O is present here, so it becomes, this bin is given a value of 1. And if it is not present, for example, another bit, which is the second bit, is not present, or this third bit is not present, so it is given a value of 0. And this ring is present here, so it's given a value of 1. So in this way, you represent the compound in the form of substructural feature, but in the form of zeros and ones. And another way of representation is extended connectivity fingerprints, where you don't focus on a fixed bit of strings, as I told you before, but you make your fingerprints based on data set to data set. And for example, it is also known as atom environment fingerprint. So you focus on a single central atom, and then you increase the radius up to a certain length, and then you find all the given substructure within that certain length. So here I'm showing you ECFP4 of bond diameter 4 or radius 2. And so the bond length would be up to 2. So it can only extend up to 2 from the central atom, which is this carbon. And therefore, all the substructures contained within this this atom environment are given a particular bit number in the bit vector. This would be more clear with the example. So I have put another example. For example, if you see this central atom and you increase the bond length up to 2, or the radius up to 2, or diameter of 4, so you get, this is the central atom in layer 1, you get this substructure. Layer 2, you get this substructure, and all the substructure which are within this layer, and you have different 32 bits assigned here based on whatever the substructure occurs here, and then you put it as on or off in the similar way as I showed you with the Max. And next you iterate it to another central atom, and you get another different atom environment, and you label it with the bits of another hash function with different bits in this case. So in this way, you get different type of bits by iterating over the whole molecule. So it is the value of ECFP gets different for different data set. So suppose if I have a huge amount of data with active and inactive compounds, so I would run this ECFP algorithm over that, and I would get a unique set of fingerprint for that particular data set. And I would not limit my research just on the 166 bits, which I showed you in the max fingerprint. So these hash values are then collected together to form the fingerprint and for a particular compound, and you can either concatenate it or so on. So you will get a fixed length of fingerprint for the whole data set, which is unique to that data set. And of course, if you want to make a user-friendly fixed length, for example, generally 1024 bits are used for the length of the ECFP fingerprint. So you can use a modulo operator where these hash values are moduled by the number of bits you want to make the final one. So you divide it by 1024 and the remainder you get is the label of that bit. However, this one is a bit tricky because you can have multiple structural assignments to one bit. So when you do predictions or so on, so you have to track it back to the original structure from these hashed fingerprints. And by the way, these hash values, the numbers I'm showing, because you must be wondering what are these numbers. These are like specific numbers based on certain values like atom number and the bond order or so on, valency and different like charge, number of hydrogen atoms. So in this way, a particular point is given a particular property value, and they constitute these hash values, which are different for different atom environment, which can be collected together or form these kind of fingerprints. So once we have these fingerprint or the bit notations of the compounds, we can find the molecular similarity, and this molecular similarity can be found with different similarity metrics. One of them is very popular, and it's called as Tanimoto coefficient, which is used very general in a majority of the similarity method. So in this way, you have 2-bit representations and you can calculate the similarity based on this formula. So for example, in A and B, you can see the blue ones are only present in A, and these are these bits which are 1 in A, and there are only B bits which are red ones which are only present in B, and then they are both A and B ones. So then basically you can represent this in this formula, and you can calculate in between range of 0 and 1 how similar two molecules are. This would tell us a large this was for example, in large database you can check with one small compound and check similarity with each molecule, and then you would know, okay, these substructures are very similar to one compound. So this is a kind of similarity measure for different compounds once they are represented in the bits form. And then we come to the concept of similarity searching. So as I told you, we can use these similarity matrix to eliminate a large fraction of inactive compound, because similar compounds generally have similar properties, as I told you before. So this is one of the concept in ligand-based virtual screening of databases, where at least one active compound must be known. Suppose this is a reference compound and you subject it to similarity with the whole database, and you find very similar compounds as analogs. And some compounds are inactive, which are not similar, which are represented as squares here. So they are not very similar, so they are inactive. So in this way, you can filter your database or search your database based on the similarity. And of course, this ligand-based virtual screening can be made in different ways. So a perfect model would be that we only rank only active compounds. So this is a database where green are active and red are inactive. So it can be either perfect way, just fetching the active compounds from the database based on the similarity, or in the random way, uh, as it is shown here. But a more realistic model, because sometimes similar compounds might differ at one small place and it leads to a large difference in the potency, which is generally known as activity cliffs in chemoinformatics. So a realistic model would be that this similarity search approach would not give all compound as active, but it would be more that some are, even it is quite similar to inactive compounds at certain point. Majority it is similar to active ones. So we need to enrich the database where the active compounds are ranked above the inactive compounds. And there are different classification methods which are used, where you can use these reference active compounds and you can enrich your database or so on. So you can assign different classes, for example, using clustering, partition, decision trees, etc. However, because the similarity option is, as I told you, that there might be false positives existing between that, because the active class is often large, because most, if even if you set, it depends on you. For example, if you set a Tanimoto similarity criteria of 0.5, then there might be some dissimilar compounds also. Or you set it as too high, then you have no active compound. So it's always a trade-off, and it's always different for different uh, project or research which you conduct. And in similar way, you can also use a ranking method for using Tanimoto coefficient. You can rank your compounds where they exist in the database, and based on the similarity. So maybe you can take a reference and find the similarity and rank where are they. And you can select an appropriate size like 1% of the database, and then you select it as active compounds. But so once you do these virtual screening approaches, you need to evaluate your compounds, what you get from these approaches in silico similarity approaches. So there can be two ways. One is prospective studies, which are the experimental evaluation of predicted compounds. So you find these small number of compounds from large number of compounds, and you take it to the lab, and then they predict under different assay conditions or so on. Of course, the tested resources are less because you already reduced the number of compounds. Or there can be retrospective studies, which are called as benchmarking studies, where you use non-active compounds, and then based on certain matrix you see, okay, which compounds I predicted as active and how much percentage of them or so on. So these benchmark virtual screening calculation work in this case. So for example, you have these active compounds and you take a reference set, which is the training set, and you use a virtual screening method. And out of these active compound, you fetch a small amount of test set and you put it in the inactive database. And through your virtual screening method, you see where these test set, which are already known to you as active, exist when you rank the database or when you uh, predict them as active or inactive. So you see the enrichment, and this is known as benchmarking, which is without the experiments. And there are certain metrics, for example, hit rate or recovery rate, that how many compounds are contained. So hit rate can be a fraction of active compounds in the whole selection set out of both inactive and inactive, and recovery rate could be only the active compounds, how many active compounds were recovered or were predicted as active. So these were the different virtual screening techniques, or a basic of virtual screening using similarity search. And however, these traditional QSAR techniques like multiple linear regression, or similarity search are generally not applicable because of the size of the data set. Because when you do high throughput screening set, the sizes are usually too large, and these data sets are usually structurally very diverse. So you, instead of finding similar uh, compounds, you are always interested to find diverse compounds, because you are looking for new compounds which are active against a particular target, or in scientific language we call it as different scaffolds, which are very which could be active against a particular target, or so on. So in that case, you need to find the patterns in the data which is contained, and not just base your calculations on the similarity, a simple similarity. And that's when machine learning techniques are used. So as you can see, suppose in a database like this database, there are large number of compounds here, and there can be so many structural patterns which are hidden, and you cannot just visually inspect these compounds and find these patterns, because it's hard for even a medicinal chemist to look into thousands or millions of compounds one by one. So therefore we have to find a way to find these patterns which might explain the data, and which can help in predicting new instances. It is similar to this example, like one is standing and lying. So you, based on the structural pattern, you either predict green as standing and red as the lying one. And in this way, for the compounds, we want to predict as active or inactive. So these machine learning techniques are used in that case to identify these structure activity relationships, which I mentioned in my initial slides also. And in this way, these compounds, some of these compounds in a database can be represented as training set which have a known label. For example, this compound has active label and this compound has inactive label. And these are the molecular fingerprints which I explained in the previous. And you test a compound with the same molecular fingerprint based on the machine learning model, and you predict it as active or inactive, because the computer has already learned different patterns contained in these compounds, and it tries to predict based on those patterns these test or unknown compound which is not known to the model as active or inactive. And in case of regression, you don't classify as active or inactive. Rather, you produce structure value relations, because you have a particular activity value like IC50 value or MIC value in that case. So you find the structure value relations. So there are different types of machine learning algorithms which are used, and one of them is like supervised machine learning, and some are unsupervised machine learning. Supervised, we already label the training data set, as I showed you. We used either classification or regression. But unsupervised, we don't label it. We cluster based on certain different approaches, like nearest neighbor cluster or so on, during which I won't be discussing in this presentation. So I would be more focused on supervised machine learning techniques. So there are different types of machine learning models, and let me first start with the random forest. So it is based on a forest of decision trees. So decision trees looks something like this. So you have one compound, and you look into each and every substructure whether it's present or not. So the compound, the model is trained based on the sequence of substructures. So suppose double bond O here is present, and next it followed by this ring, and then the benzene ring. So if all these substructures are present, or the combination of substructure are present, then it is either predicted as active or as inactive. And suppose a different substructure is present here, then it is predicted as inactive. So in this way, there are a set of formulation of rules in a particular decision tree. And this is generally trained from the random subset of training data. And you construct many such trees, like an ensemble of decision trees. And then you have for example, hundreds of decision trees, and then based on the majority voting of a test compound which is subjected to all these decision rules. So out of 100 trees, suppose 75 are predicting as active and 25 as inactive. So we focus our prediction on the majority voting and we predict the final class of the compound. So this is how random forest works. Another machine learning model which is popular and also traditional is support vector machine. So in this support vector machine, you project your training data into reference space. So for example, two-dimensional space here, and you derive a separating hyperplane between the positive and negative training data. Suppose this is the positive class or the active and this is negative class. So you try to separate the data based on some mathematical calculation, which is like hyperplane maximization. So you can imagine that the active class and inactive class is projected in two dimension, and there is this single hyperplane which is separating it, and this particular hyperplane is chosen, and it is maximized. The margins are maximized for this particular hyperplane till it best separates the two classes. For example, you can see the red ones are the margins here, and these training compounds which are present on the margins are known as support vectors. And this is why this method is called a support vector machine. So it is based on some mathematical formulation where these active compounds you make a hyperplane and you maximize it based on a normal vector and a bias. So this is how it looks, the mathematical form, and which has a scalar product here as written in these brackets, and you maximize these hyperplanes. So I won't go into the mathematical detail, but this is how it works. And but there is a catch here, because this data is linearly separable. So it's written on a paper and you can separate it easily, but not all the data is linearly separable. And sometimes we have non-linear data, which is, for example, this one. There is no chance of finding a hyperplane between which best separates the two classes. So in that case, you project your low dimension data into a high dimension, for example, 3 dimension here, and that in 3 dimension you find a new hyperplane in that feature space, and this hyperplane best separates the two classes here. So for example, this is 2 dimension one, and then you project it based on mathematical function with 3 dimension, and then now you can see the red ones are separated from blue one. And this is done with the help of a kernel function, and these kernel functions are represent the scalar products which I told you in the hyperplane equation as in the higher dimensional space. So you explicitly project your data into higher dimensional space. It has both advantages and disadvantages. Advantage is good for the non-linear data, you get a good separation. However, the calculations become complex in that case, but generally the data is very complex and it's non-linear in that case. So you can either do the binary classification. So if you have a value predicted as 1 of this decision function or from the hyperplane equation, so you predict it as active, and if you have fx as -1, then it is predicted as inactive. And these decision functions contain this kernel function which I told you, and it depends on the support vectors from the hyperplane. Also, you can do a SVM ranking, like I showed you the virtual screening ranking based on similarity search. In similar way, you can get a value based on, that is the distance from the hyperplane. So more the distance from the hyperplane, and its ranking will be towards the inactive side. So in this way, you can rank your compounds and in a way do a virtual screening using these machine learning models. And there are other machine learning models also which are very popular these days, such as deep neural network. which consists of hidden layers, and each layer comprises of hundreds of nonlinear process units. So it can take large number of input features and neurons and can automatically extract features at different hierarchical levels. So this is a deep neural network with 3 hidden layers. So there are different functions and in each layer where you transform the data as well as you predict to the final output layer. And in this way, this method is also very popular these days. Apart from, there are many different types of this method which are available. And now the model evaluation. So as I told you, you get a compound ranking or so on. So based on the active or inactive, there are several methods of model evaluation. One is the receiver operating curve, which I think majority of people use these days, which is a graph between the true positive versus the false positive rate. And its random value is 0.5, which is the diagonal here. However, the more it moves away from the diagonal based on the active and inactive compound, it shows the prediction accuracy increases in that case. So this is called area under the ROC curve, or AUC value, which is more, if it is more than 0.5 and more towards 1, then that is the perfect selection. Optimal selection is the 1. And this kind of is a metric for the ranking of compounds. So after discussing all these different aspects of chemoinformatics, I now want to focus on 2 case studies or 2 projects which I have been involved in. One would focus on the regression, that is prediction of antifungal activity against a fungi species called Aspergillus fumigatus. So in this project, I focused on because this Aspergillus is a very common fungi, and it is also very dangerous because it is causing lot of infections in COVID-19 related patients and so on. So therefore, this disease was the prime focus because of limited amount of drugs available for this Aspergillus species. And therefore, I conducted a in silico protocol for this disease. And there are different mechanism of action which are given in this paper, which I showed you, which focuses on finding novel antifungal agents and their activity against Aspergillus species. And there are different, but mostly their classes are like azoles, etc., which are more popular inhibitors or more popular for the use against Aspergillus species. And these fungal infections, as I told you, are a cause of high mortality rate worldwide. And this disease caused by Aspergillus species is known as aspergillosis. And there are limited drugs in the market, and they are based on a particular classes only, and they have toxicity, drug-drug interactions, and so on. So I wanted to conduct an analysis based on the diversity of the data I have, and therefore I looked into Campbell database to search for new antifungal compound against Aspergillus using computational technology. And after doing some literature survey, I found that natural products are kind of very useful source for as antifungal agents, and they have proven to be potent antifungal based on their easy synthesizability or so on. So I tried to search, and I'll show you a virtual screening protocol for this particular project. So my aim was whether computational models can learn structural or physiochemical information embedded in already existing anti-Aspergillus compounds. So I wanted to see, because there are different, how diverse set has been tested against Aspergillus before, because most of the drugs which are in the market are azoles or so on. So therefore, research had it motivated me to research on the diverse set of molecules. So I used my aim was to, can in silico model be used for the prediction of new compounds having antifungal activity against Aspergillus. and which databases could be good source for screening these new compounds. So therefore, I looked into the Campbell database and I found activity against this Aspergillus species, which is like I found organism-based assays related to it, not direct protein-based, which is like chitinase or so on. And majority of the data was contained in organism-based, as you can see, and very few was available for like chitinase or certain kind of enzyme. So I focused on this data and I looked into the activity measurements. So majority of them had MIC measurements, which I discussed in the first initial part of my presentation. And therefore I looked, generally it is known that if MIC is less than 1 microgram per ml, then they are active against that particular microorganism. So I took this data and I try to pre-process the data, because it is also very important for a project in chemoinformatics to first of all refine and curate the data, because the data is in a very raw form generally. So I used this data pre-processing and I selected compounds with MIC values, and I used only the standard relation of equal to, and I used like molecular weight less than 300 Daltons. I used this because I only wanted to focus on the small molecules, and generally because the drugs are usually small molecules which are orally acceptable or so on. So I took this criteria, and of course I disregarded ambiguous measurements or so on. And I washed and canonicalized using different softwares available for like OpenEye or RTKit, and which are usually available for processing the compounds. And also I removed potential liable compounds using certain filters and so on. And therefore I was left with 447 unique compound data set after pre-processing the data. And this is the distribution of these 447 compounds in terms of MIC value. So you can see 59 compounds have MIC value less than or equal to 1 microgram per ml, but majority of them had MIC value high. So they were tested, but we cannot say that they are active, but we had the MIC value for them, which could help us in making the models for MIC prediction. Of course, MIC can be very large, as you can see, 10,000 or so. So to standardize the data and for making a better model, I rescaled MIC values in using log, and I call it PMIC, so as to scale the data or normalize the data in this case in a usual form. And then I use the descriptors, which are my fingerprints, of course, as I told you, like different types of fingerprints. So ECFP4 I already discussed in this presentation. However, there are other types of fingerprint too, like Morgan fingerprints, which are based on the Morgan algorithm. And it is also kind of similar to ECFP fingerprints, but with a certain radius, like radius 2, and sometimes they are count binary Morgan fingerprints, because you, instead of giving the value of 1 or 0 to the bit, you give the count of the substructure which is occurring. And you can fold the Morgan fingerprint, as I told you, the hashed fingerprint, where multiple substructure can be related to one particular bit. This will reduce the calculations or the efficiency of the model in that case. Also, I used different property descriptors from a software called Molecular Operating Environment. And there is a popular, not fingerprint, but property vector available, which is called as mol2vec, where you convert your molecule into a set of strings of different properties or so on of different radius. So it's similar to word2vec in natural language processing. And then you have to still pre-process the data, because there might be highly correlated variables in these fingerprints which should be removed or which have a threshold of over 0.9, because generally more correlation lead to bad prediction, and in it would not explain the data very nicely. So I used models. First, I used remove the highly correlated features, and next I used assembled models to calculate correlation between X and Y, and deleted variable with no correlation to Y, which doesn't explain Y with respect to X. And there are different type of regression models like least square, partial least square, elastic net, random forest, and support vector regression is also used, but there are other methods like based on the Bayesian process, Gaussian process, etc. And different kernel functions can be used for SVR like RBF, or there is a kernel function based on Tanimoto coefficient, which is called as Tanimoto kernel. And again, like AUC, ROC, there are different regression model evaluators like coefficient of determination, which is called as R square, or mean absolute error, or root mean absolute squared error. And it tells how the predicted values deviate from the original values, and generally the error should be the low on the lower part, and the coefficient of determination should be on the higher part towards 1, which tells us about the goodness of the fit of regression models. So these are some of the results which I got for this data, and for ECFP4, I generated around 2368 bit vector, which is the unfolded one. And after the feature selection protocol, I could get 225 bit vector, which I used for training as well as test. 80% of the data was used as training and 20 as test. And then I used 5-fold cross validation. So by 5-fold cross validation, I mean that I made this 80 training and test split, 20% test 5 different times, so that each training set should not be used as training again in the second fold of cross validation or so on. So this is how we generally predict the efficiency of the model. And these are the results. The first row shows the training result and R square, or the test result, on the second row. And you can see certain models worked really well, like linear SVR or ridge regression and Gaussian process with R square value of 0.8. I used other models or with different descriptors. However, they could not perform as good as ECFP4. So I stuck with ECFP4 for my model, as it could explain or predict the data well. And these are like Y-Y plot difference between actual Y and predicted Y. And you can see they are mostly towards the diagonal, which means what the actual Y was very close to the predicted Y. And these are different error measurements which were on the lower side. Also, as I told you, I was looking for diverse compounds. So therefore, I looked into the Tanimoto similarity matrix, and which is the data diversity matrix. And you can see I plotted all the compound. This is just to visualize 100 versus 100 compounds, but I made for the whole data set the diversity matrix, and I could find you can see this value of Tanimoto similarity ranging from 0 to 1. So darker means they are very similar, but it's more on the lighter side here, which means that they were not very similar in terms of the ECFP4 which I constituted, which means the set was quite diverse. And to further do rigorous validation, you can make scaffolds for different compounds on the data set. So you can get unique BM scaffold for the data set, and this is the distribution of scaffolds. So scaffolds are generally generated after removing linkers or so on, so that the core part of the molecule is conserved. And if the core is very different, as you can see, most of them had unique 186 unique number of shared compounds were 1 between 108 scaffolds. So it means that the data set was very unique, because not many compounds shared common scaffolds. So I also did rigorous validation in this case by doing scaffold based splitting, which is like 80% training and 20% test set based on different scaffolds, so that scaffolds in the training set are different from the test set. And in the similar way, I performed the 5-fold cross validation. Although you can see the performance decreased and it reached 0.70, but overall, still it was quite high, which means the model which we built was quite good in predicting diverse compounds too. And also using these regression techniques, you can find which bit contribute higher towards the prediction. So these are like a graph of regression coefficients, and you can see these bits which are showed here are contributing negatively and positively towards the prediction. And you can see these are labeled on the compounds, and this bit 300, when it was present, it means that the MIC value was reduced, and when this was present, it means MIC was increased. So you can look into these. Even the similar ones were obtained from ridge regression model, and when I looked into these substructure into compounds with MIC value, so this bit 300, as you can see, it was mainly contained in compounds with low PMIC value, and this was with high PMIC value. So it was only contained, and it was not contained in the other one. So these were on the opposite side. So these could constitute very important substructure, which can further we can look for constituting virtual compounds in future for them, so that which we can look into these substructures in detail for constituting compounds against Aspergillus fumigatus. And then the final model, because the model worked well, it can be used for virtual screening, and we can select the best model, which was with the ECFP4 fingerprints, and we can train using on all the selected compounds with the 447 compound, and use these model for screening a database. So for example, as I told you, natural products can be good source for Aspergillus fumigatus, and therefore I use Dictionary of Natural Product database, which is a commercial database, and I curated this database for prediction of compound, and therefore I converted it into the similar 225-bit fingerprint, and I screened this database to find some 415 compounds that had PMIC value of more than 6, which means MIC value less than 1 microgram per ml, based on the model we constructed. So these compounds could be a very useful source. So we reduced testing so many compounds into a smaller set of compounds, which can further be validated based on the substructures which I showed you, and also based on, if we know, for example, chitinase is an important fungal cell wall enzyme. So we can try to do structural base work of these compounds on chitinase and try to validate it further whether they are good potent compounds against Aspergillus fumigatus. So this was one of the protocol which I wanted to show you about how the virtual screening protocol is done using chemoinformatics or regression models. And lastly, I want to quickly discuss another case study where I don't use the regression model, but I use classification model, that is machine learning models. And this project is another project where I focused on assay interference compounds, which are called as PAINs and are generally used as filters. So these pan assay interference compounds are small molecules that can produce false positive assay signal due to reactivity under assay conditions. So they are generally classified into 480 compound classes, and people use it to filter their data for liable compounds that can frequently hit or can cause false positive signal, such as due to different mechanism of action like autofluorescence, etc. So paints are used as filters to flag compounds with potential liabilities, and these were found in 2010 by Bell and Holloway when they conducted a certain set of experiments on these reactive classes which are contained in the compounds. And they are used as filters, as I told you. So if it is detected, like this red one is detected, and therefore we don't use this compound for further optimization, but we can, the one compound which passes the Payne filter and do not have any of the 480 classes of Payne's filter can be used for further analysis, as it is not liable compound. This is how these Payne's filters have been used before. And these Payne's classes are different types like quinone, catechol, rhodanine, and there are various mechanisms, as I told you, like autofluorescence or covalent modification of proteins, sometimes metal chelation or redox reactivity, and this make them like frequent reactive compound. However, there is a catch, because in one of the study I performed during my PhD time, I found that paints also have a diverse activity profile. Like you can see these two compounds here, they have the same quinone substructure labeled in red, but you can see, and they are tested in large number of assays, but one is frequently hitting, which is showing activity against 70 of them, but this is showing activity against 0, so that means it is inactive. So this is generally known as dark chemical matter, because it is tested in large number of assays, but still it is inactive. And they have same Payne substructure, and they had a large number of common assays, 187 common assays, 90% of. Still you can see the difference. So it's not the Payne substructure which make it reactive, but also the surrounding substructure, because it's the structural context of Payne's which depends whether one compound would be frequently hitting or not. And if it is frequently hitting, we generally call it as promiscuous, but obviously in a false hitting, false positive perspective, because there are also promiscuous drugs available which are in a good way, but this is in terms of false positive hit. So therefore, what I did in this case, because it was important to not only just rely on the Payne's filter, but also rely on the structural context of Payne's. So what I used is I constituted data set using promiscuous and DCM, which is dark chemical matter, and try to use machine learning to identify certain patterns or the structural context which is used on these Payne substructures. And as you know, in the machine learning set, I made active set, inactive, active constitute promiscuous Paynes, and inactive as DCM Paynes. And for this purpose, I needed a extensively assayed data, which I got from PubChem database. As I explained you before, it has a large number of extensively assayed compounds which are tested. And therefore, I use this data set, and around 85% of these compounds were tested in more than 100 assays. And different type of Payne's filters are available in different chemical repositories like RDKit, Zinc, and Campbell. So I applied all these because sometimes implementation discrepancies occur. And therefore, I found around 27,000 compounds which were detected as Payne's, which contained around 270 Payne substructure out of 480. And these data sets were constituted first, and then based on these extensively set, which were tested in more than 100 assays, I divided my data set into two parts with more than hits, more than 10, and number of hits equal to 0, that is the inactive one. And you can see I got 5,000 compounds as frequently hitting ones and 3,000 as inactive. And they had around 74 Payne substructure which were common between these two classes. And therefore I focused my research on the common ones, as I showed you an example of a common one, because I want to explore the structural context around that common substructure. And looking into the number of promiscuous Paynes and DCM Paynes in these 74 overlapping classes, you can see I divided the log of promiscuous panes and DCM panes. The distribution shift towards the right hand side, and as it has this shows that there is a data imbalance. There are more active panes than inactive ones, so I need to balance my data, and this is also a very important uh, aspect in machine learning, because you need to balance your data. If the data is imbalanced, your prediction can be biased towards a certain class. So for example, you have this panes class called Manish base, and you have around 327 promiscuous panes and 108 DCM panes. So what I did is took the minimum of the two, and I randomly sampled these number of compounds from promiscuous panes, so that I have equal number of active and inactive compounds. And then I divided equally into training and test. So training contained 54 as active, 54 inactive, and the test also contained 54 and inactive and active. So 50% split I did. And I used only those panes classes which were represented by at least 2 promiscuous panes and 2 DCM panes, so as to balance my data set. And these 10 independent sampling trials were carried out with 10 balanced training and test set. So each balanced training and test set consisted of 1900 compounds, which were enough number of compounds for doing a machine learning analysis. And these are the different metrics which I used. So as I told you, people generally use AUC ROC curve, but there is also called a metric available named Matthews correlation coefficient, because sometimes when your data set is imbalanced, so you need to use MCC, because it takes into account, it gives good predictions on the imbalanced data set compared to AUC and so on. Because if you have majority of actives in AUC, then sometimes what happens is they are all predicted as active and you have very good result. But still we balance the data and also use 3 different metrics to be sure about our predictions. And then we use 3 different models. One was SVM, random forest, and deep neural networks. And these are the result. As you can see that AUC with ECFP4 was quite good compared to MAX. And all the 3 models almost performed quite well, but the traditional ones like SVM and random forest were performing quite good. And looking at the AUC, ROC, MCC values, you can see AUC, ROC reached 0.9 for all the methods, and MCC also was close to 0.70, like 0.65 or so on. So this means, and the standard deviation was also less, which means that most of the predictions were consistent enough. So therefore, the models which we built were quite acceptable, because they predicted quite nicely the structural context of panes. However, still, because the predictions are done by the computer and machine detects it in the form of zeros and ones, but to go to the next step, we need to know whether the predictions are chemically intuitive or not. So what we can use is, we have certain methods for feature interpretations, like which features are important for particular prediction or so on. And one of this method, because SVM performed quite well, so I used Tanimoto kernel, which is based on Tanimoto coefficient, so the kernel function which I explained before. And I used this method because it had been published before in my previous group, and I use this method for weighing the features of the compounds, like which are important for the prediction and which are not important for the prediction, and for towards positive or negative. And we use this formula for that case, where X is the test instance and Xi is the support vector. And these are the support vector coefficient which we get from the mathematical equation of SVM. So it would be more clear with an example. So suppose you have a test compound and you have 3 support vectors, or the training set. So you try to find the kernel function. So you can see it has 3 bits on and it has 2 bits. and there is one common bit between test compound and the first support vector. So the kernel function would be based on the Tanimoto formula. So you have 2 plus 3 bit on minus the common bit, which is the 1. So the denominator becomes 4. And 0 by 4 means if the first bit is common between the two. So it's not common, so it's 0. If it is common, then it is 1. And in this way, you can find the kernel function value. And for feature weight, you constitute all the first bit values, and you multiply it with the support vector coefficient, which is lambda y and so on. So lambda 1 by 1 are for support vector 1. So therefore, you get these values, and you find the feature weight of a particular feature here, bit 1, and this would tell you how important this bit is for the prediction of the model. So in this way, I found for all the test set which I had, like 1822 compounds, I found these feature weight matrix, and if you sum up the column, you would get feature like whether it is predicted as active or inactive. So it's that way. So therefore, I just summed it up on the row wise, and I tried to find whether because some are always on the positive side, some were always on the negative side. So I found around 2573 positive features and 2527 as negative features. And these contributing features, some of them, the top 31 based on the sum, were uh, visually inspected using with the help of a medicinal chemist, and we could find some chemical intuitiveness like this example. So these are the for enrhodinine class, 3 positive features are mentioned here, and you can see that if they are like, as you can see that this feature which is present on the exocyclic double bond, and it is generally known to be reactive based on the Michael acceptor activity. So it generally a nucleophile attaches here, and therefore it leads to like a thiol comes here and reacts with this point, and it leads to high reaction in different assays. So this was kind of chemically intuitive with feature interpretation, whatever results we got. If chlorine occurred here, that means that with the presence of chlorine, this electrophilic nature of this double bond increases. So therefore, a nucleophile can attack it more easily. So these structures which we found were quite important, and they were chemically intuitive. So in a way, feature interpretation could rationalize our prediction in terms of chemistry or so on. So these were different conclusions which I got from these research, that using I could construct high predicting accuracy models, and I with machine learning could exploit the structural patterns responsible for pain's activity or inactivity. And machine learning extends the capacity of pains. We don't not only rely on the pains filter, but also on the structural context of the pain or the structure surrounding the pain substructure. And the highly weighted positive feature which I got could be associated with the reactive moieties, as I showed you in the last slide. And therefore, still, because the predictions are done from machine learning, so not all prediction could be rationalized in terms of activity, but still we could relate some of them, which was quite good. And this research, I was able to publish 2 papers in Journal of Medicinal Chemistry. One was focusing on data mining, and one was focusing on the machine learning models which I generated and their chemical intuitiveness. And this was uh, uh, a good success I got in this research project, and I look forward to work on more such projects in future. And this is basically the people I want to acknowledge who have worked with me over the years, and my guide in my PhD, Professor Dr. Jürgen Bayroth, from University of Bonn, Germany, and the group currently I am where I have Professor Dr. Kimito Funatsu and Associate Professor Tomoyuki Miyao in NICE, Japan. And also a postdoc whom I worked very closely with, Dr. Yehu, who was very much important factor in publishing these research or so on. And this is my current group in NICE, and we do chemoinformatics and different aspects. Some do structural based drug design, some do quantum mechanics. So a diverse group, but we look for interdisciplinary projects, and we also look to uh, do more research and exciting things related to chemoinformatics. So this is my presentation for today. I hope I motivated some of pharmacy people to join chemoinformatics in future, and because chemistry knowledge with computers is quite exciting, I suppose. And yeah, so I look forward to your comments and questions. Thank you for your kind attention.
Thank you very much, Dr. Swarit Sensei, for the interesting talk. I know you are very busy because I know it for sure, but you've done it very well, and the talk is very interesting. And as Swarit Sensei said, please, if some of the pharmacy student really interested in joining us, please join. So, okay, we are coming to the questions and answer sessions, and you can ask in the chat box, or you can raise your hand. You can ask in English, or if you are too shy, you can ask in Bahasa Indonesia, or maybe in Germany, but I can understand Germany. So, okay, there is one question in the chat box from Fikan RSPSS. Thank you for your interesting explanations. I would like to ask, how do you select the features, which is 225 bits in the first project? Is it from trial and error in the model by selecting the best evaluation numbers?
Okay, thanks for your question, Vikan. And basically, I explained it that I used ECFP4 fingerprints, and for the whole data set, I generated fingerprints. However, I use this feature selection protocol, which is mentioned here. And so I get a certain number of ECFP4, as I explained, 2368 bit vector initially. So a bit vector which had all the features of 2368 and number. And then with the help of feature selection protocol. So generally in these regression protocols or machine learning protocols, we remove the highly correlated features initially because they can tend to bias the model and predict quite well because they are very much correlated with each other, or because we want a diverse set of variables which can uh, find the patterns, like diverse set which are related to each other, and kind of their combination predicts the regression value very nicely. So therefore we deleted the highly correlated values, X variables initially, and then based on 3 different models where I used lasso, and I used linear SVR and ridge regression, which I didn't mention, but I just mentioned 3 models at the same time, and I deleted that variable that had no correlate, that does not explain, that cannot predict Y variable very nicely. So it had no correlation to Y. So this is one part is correlation between X variables, and one is correlation between X and Y. So it's 2 different cases. We don't want correlation between X, but we want correlation between X and Y so that it explains the data well. So this is how I reduced my ECFP4 feature to final 225 bit vector, which I got I hope I explained. Thanks. Rezi, I cannot hear you.
Yeah, sorry.
No.
Sorry. Yeah, thank you for your answer. And yeah, I think is there any other questions? I will also like to extend a very nice welcome to our Dean, Professor Ari Anwar. He just joined.
Yeah, hello.
Hello.
Professor Aswaf Jasyal, yeah?
Yes, I had.
Very
Well,
nice to
nice
see you here. And also to Mr. Rezi. Yeah, long time not to see you. So I hope can visit nice and
Yeah, please come.
near.
Yeah, sure. We are looking forward to your visit in future.
Yeah.
Yeah, so Professor Ari is my supervisor and doing structure-based drug design before.
Ah, nice.
So,
Yeah.
yeah.
Very interesting about your work about pains prediction. Yeah.
Yes, sir.
Yeah, because pains is also resulting the false positive. Yeah. So
Yes.
using the prediction, we can avoid using some result with
Yes.
false positive. Yeah.
Yes, because paints are generally used. There are many different filters which we use, like Lilly, Metcalfe rules, paints, and there are other things which are used. For example, they coagulation or so, like aggregators are very popular these days. But for pains, we found that they can be inactive also, or can be less frequent hitting ones. As I showed some examples here, these are less frequent hitting, so they can become an active molecule or a potent candidate in future. So we cannot just filter them out just because they have pain substructure. Rather, we should look into the structural context of the pains. What are the features which are around pains, and if they are, if because using machine learning models, if you have good predicting models like I showed you, so you can add an additional filter to the pains compound, and then you can filter those ones which are frequently hitting, then those ones which are inactive or non-frequently hitting. Then this is interesting because then even pains compound can become a drug in future. Yeah, so it's kind of interesting than just removing the compounds based on the pains filter. So yes, of course they are useful filters, but we cannot rely on them fully because we need to explore the structural context around the pain substructure.
Yeah. Okay. Thank you. Yeah.
Thanks for attending.
Yeah.
Thank you, Professor Ari, for your questions. And we are waiting for you here. Maybe in the future for the collaboration.
Yeah.
Okay. Is there any questions? Any other question from the audiences? Okay.
Can I ask questions?
Yeah, please.
Okay. Thank you for a very, well, I may say very challenging presentation actually, because it's not really my expertise, but I have questions about the structures or the chemical complex of the compound. Is there any fundamental difference between chemoinformatics for synthetic compounds and compounds isolated from natural products? Because, well, as you know, like phytochemical structures are usually pretty complex and they're pretty big. Is there any restrictions for the complex regarding how big they are or how complex they are? Thank you.
Yeah, thanks for a nice question. So I encountered this problem because when I was curating Dictionary of Natural Products, so because it has around 300,000 compounds, Dictionary of Natural Product, and when I curated it, as I showed you in the slide, I've written it curated previously from DNP database, fragment like natural product. So I divided my data into two different databases basically. One is the fragment like, which are small molecules, but majority of them out of 3 million, I could only get 50,000, which are fragment like. Majority of them are big molecules with large, very complex, and they have molecular weight like 1,000 Daltons or so. It's a big molecule. And for generally the databases I showed you, and in chemoinformatics, we mostly focus on small molecule drug discovery, and not because they are generally small molecules are orally absorbed, they can be absorbed, they have good pharmacokinetics properties, but a large molecule cannot be disintegrated or so on. So we cannot just focus our research on the large molecule. But of course, it is an interesting aspect, and I plan on doing certain things with it, like focusing on small parts of big molecule, which might be interesting in generating analogs for a new analogs for some already existing part. Like we can get a different source of a compound from a natural product, which can be synthesized, which is easily available from a plant or so, like which can be used as analog compound for fetching like a small part or small core of the molecule. And this would be interesting aspect for chemoinformatics, but I haven't seen any research and I haven't done any of that thing yet. But yes, of course, they are more complex and cannot be used directly. That's why I use these molecular weight criteria of 300 Daltons or maybe different parts. So yeah, I mean, it's an interesting aspect which can be explored in future. Yeah. Thanks a lot for your question.
Thank you for the explanations. Very clear.
Yeah.
Thank you for the question. Yeah, I think it is an existing problem because we usually use natural product compounds in many Indonesian research. So yeah, it will be interesting to see about that.
Yes, I mean, owing to their synthesizability and availability, natural products are still an interesting topic since last 20 years or so. And even in NICE, like Professor Kanaya's group has this knapsack database and different. So I've tried to use that too, but they are manually curated. So a lot of work has been done on like curating the database, but obviously a lot of unanswered questions, and especially because we mostly focus on drug designing and natural products, complex ones, how they can be used in small molecule drug discovery is still a question. So maybe we should look into it in future.
Yeah, thank you for your added explanations. So is there any other questions from the audiences? No, I have some questions myself. We know that data is growing. As we talk, the experiment around the world is being done and it is quickly growing. So I want to ask how impactful new data sets so that we need to redo the calculation of our models. Say that do we need to redo our calculation every time a new data set comes, or what do you think about it? Thank you.
Yeah, I mean, again, this answer to this would be depending on project to project, because it depends what you are doing, what is your research topic. For example, you do structural based drug designing and you focus on a particular target in that. And of course, some targets will have very few compounds, some will have very large number of compounds. And so it's always a question, what is the sufficient data for a particular analysis? Yes, we want to find the best ones and we want to explore as much as possible whatever chemical space which we are provided with. But maybe in certain projects, when the chemical space might not change and no new structural diverse compounds are available, or maybe we have reached the saturation level for a particular target, like kinases are so much these days, so much data is available for kinases. So I don't think there will be any change in future in kinases. Maybe there is, but so far it doesn't look like, because whatever we saw the research or read the papers are kind of similar now. There's nothing, no, but maybe it's an interesting topic, whatever map
Ada Pak
we
Rezi,
found.
guys.
And next, I want to say that yes, data is exponentially increasing, and I mean, it will increase, but again, my answer would be, it's not, we cannot just say, it depends. If you search a new research idea or so, maybe new compounds would be beneficial in that. So if there are more structural diverse compounds come to your structurally uh, your, for example, a particular target you are working on, then maybe your results will change, of docking also, maybe, who knows, right? So it's the same way for machine learning. Whatever, maybe I'm making the models and I'm predicting, but maybe an entirely different scaffold is discovered, and it's but of course with the data is useful, because majority of the patterns which we can't understand, maybe machine learning can, or any other thing can. So I think it's answer is yes as well as no, so it's not a definite answer to your question. Yeah.
Okay, thank you very much. Yeah. Now I think there is another question, but maybe because of the time, maybe this is the last question. So I will read. Dear Dr. Suarit Jasiao, thank you for the interesting explanation. I have questions regarding the application of chemoinformatics. Is it possible to predict the synergistic effect or simulate the drug combinations in some disease? Thank you.
Okay, so as far as I have read some papers, or people who have worked with me in past, they did work on the synergistic effect of some drugs. For example, for the cancer cells, I have seen work where you need combination therapy, or you need two different drugs to act on different targets in, for example. And sometimes when they are synergistically important, then it leads to killing of cancer cell. And sometimes when one is going, other is down. So yes, I mean, it's an interesting topic, but obviously many, it depends on the disease you are working, because cancer is super complex. It's not just two targets or three, or there are many mechanisms involved in that, many things which are need to be taken into. So you have to design your protocol for chemoinformatics also. I mean, of course it can find a way, and you can use two different drug combination and try to predict their activity or their target identification, or you can use cell based assays for that, or so different kind of data is available. So I think it is possible to predict, and with whatever data we have available. Only thing is, it depends on the complexity of the disease and so on. So you have to take that into consideration.
Okay, thank you for your very compact answer. So I think it's time for me to end this session. Once again, thank you very much, Dr. Swarit, for your talk. And I hope we can meet you, not non-virtually, someday in the
Yes,
future.
yeah, hope
So,
to. It was a good opportunity. Thanks for giving this opportunity to me and to present in your university. And yeah, I hope I motivated some of the people for chemoinformatics in future. That was
so.
my aim. And I thank you very much for attending, each and everyone. And I look forward to maybe collaborate, meet in somewhere in future. Thanks a lot.
Thank you. I will give back the screen and time to the committee. Thank you. Sorry, your microphone still off, Mrs. Dana.
Okay, I'm sorry. Thank you, Apotek Rezi, and once again, thank you, Dr. Swarit, for the impressive and also insightful lecture and discussion. Before we move on to the next session, I would like to greet once again our honorable guest, Professor Dr. Ari Yanwar, Magister Science, the Dean of Pharmacy, the Dean of Faculty of Pharmacy, Universitas Indonesia. Thank you for joining us, Professor. And I would like to inform all participants that our committee have shared the attendance list as well as the evaluation questionnaire in the chat box. Please kindly spare a minute to fill the attendance list and also the evaluation questionnaire. For appreciations, the committee would like to give Dr. Swarit and Apotheker Rezi an e-certificate. The certificate will be given virtually by one of our committee members. This e-certificate will be given by Professor Dr. Apotheker Ari Yanwar, Magister Science.
Okay. Okay. Yeah. Thank you, Professor Swarit, for your very kind lecture. Yeah. I hope.
Yeah, thank you very much for having.
You're sharing to our student on all students in Indonesia, maybe also abroad, will yeah, strengthening the perspective about cheminformatics. Yeah,
Yep.
thank you very much.
Thank you very much, Professor, and thank you for having.
Hope we can collaborate in the future. Yeah.
Yes, definitely. Thanks a lot.
Okay, thank you very much. Yeah, this is a certificate for you virtually.
Yeah, it's okay. That's new normal. So thanks a lot.
And also thank you, Mr. Rezi. Yeah.
Yeah.
PhD candidate. Yeah.
Yeah. Amen.
Hope your research will be running well.
Amen. Also, please come to Japan.
Yes, of course.
Prospective student who want to learn cheminformatics.
Okay.
Okay, thanks a lot. Thank you very much.
Bye.
Thank you.
Bye. So have a nice day, everyone.
Nice.
Thanks for attending.
Thank you.
Thank you.
Ladies and gentlemen, now we are going to take pictures for documentation. Would you please turn on the video and please one of the committee to guide this photo session?
Okay, I will take a picture for documentation. In 3, 2, 1, smile. Okay, once again. 3, 2, 1, smile. Okay, thank you.
Okay.
Okay, thank you very much. Ladies and gentlemen, once again, we would like to remind you to fill the attendance list that we've shared in the chat box. And for your reminder, the next lecture will be held tomorrow on Thursday, 25th of August, 2022, with lecturer number 7, Professor Dr. Apotheker Aryanwar, Magister Science, from the Computational Laboratory Faculty of Pharmacy, Universitas Indonesia. That will start at 10 a.m. Jakarta time. The Zoom will be open 30 minutes before the lecture starts. Finally, we have come to the end of this event. May all of us get the value and benefits from today's lecture. Thank you very much for your kind attention and participations in this summer course. I'm Donna, signing off, and see you tomorrow. Wassalamualaikum warahmatullahi wabarakatuh.
Salam.
Waalaikumsalam warahmatullahi
Bye.
wabarakatuh. See you.
See you.
Thank you.
See you.
Bye.